Thank you very much for the invitation. Just highlighted my objectives here. I'd like to, hopefully you'll be able to know how to explain how genomic architecture leads to copy number variants and genetic disorders. And I'll be listing 7 recurrent CNVs that are associated with schizophrenia. And finally, I'll be describing how next-generation sequencing approaches can be applied to psychiatric genetics in general. So the overview of the talk, we'll start with risk factors and genetics of schizophrenia, DNA copy number variation, some of the recurrent mutations now associated with schizophrenia, rare copy number mutations in candidate genes, and then our approaches to improving copy number detection resolution with next generation sequencing technology, and finally some of the approaches we're taking to identify smaller variants with, again, next generation sequencing approaches. So schizophrenia is a severe chronic and disabling brain disorder. It has both positive and negative symptoms. The positive symptoms include unusual thoughts or perceptions that can include hallucinations or delusions. The negative symptoms tend to be cognitive and represent a loss or decrease in the ability to initiate plans, problems with attention, some memories, and executive functions. Onset is typically in late adolescence or early childhood, and the frequency worldwide is approximately 0.5 to 1%. A number of the risk factors for schizophrenia include older paternal age and gestational exposure to maternal famine, and finally family history. This is data from a birth cohort in Jerusalem, and over a course of 12 years, the births of 90,000 babies were monitored in Jerusalem, and parental age and demographic information was taken at the time, and these patients, or these children, were then followed through the course of their life with respect to diseases. And you can see here at age 30, risk begins to increase for developing schizophrenia along this axis here, and increases up to age 35 in both maternal and paternal ages. And then, obviously, just the paternal ages beyond 40, the increase goes up to approximately 2.5 relative risk for schizophrenia in fathers age 50 or more. This data has been duplicated in a number of other studies, and it's likely that there's a new supply of mutations from the increased rates of mutation in sperm germ cells, male germ cells. So the risk factor, the second risk factor is gestational exposure to maternal famine. And the evidence for this comes from 2 studies. The first is the Dutch hunger winter famine of 1944, where the German army had barricaded a lot of cities in the western part of Holland, and essentially created a famine by regulating the food supplies going into the cities. And in the fall of 1944, the worst part of the famine, the daily caloric intake was about 500 per person. So obviously there were a vast number of congenital disabilities and malformations associated with births during that time. But the patients were compared to another cohort in another part of Amsterdam that wasn't, or another part of Holland that wasn't subjected to famine, and there was a 2-fold increase in the onset of adult schizophrenia for that birth cohort that was conceived during the worst part of the famine. The same phenomenon was seen in the Chinese famine of the 1960s. This was the result of the failure of the Great Leap Forward, where it's estimated that approximately 30 million people died. And again, there was an approximate 2-fold increased risk for developing schizophrenia, adult onset schizophrenia, in that birth cohort that experienced the maternal famine. And the final risk factor I'll mention is family history. Twin studies estimate heritability at approximately 80%. And the risk is increased approximately 10-fold in first-degree relatives. But despite this, most cases of schizophrenia are actually sporadic with no family history. So what are the current approaches and old approaches to identifying genes involved in schizophrenia? In the early 90s, there were a number of linkage studies with high-risk families, and there was a lot of excitement in the early 90s with genes mapping to chromosome 1 and chromosome 5, but unfortunately the relevant mutations were never identified in those families. Following linkage analysis, there were a number of candidate gene-based approaches where common variants in biologically relevant candidate genes were compared between individuals with schizophrenia and controls with a normal mental health. And the results are pretty variable from that period. More recently, there have been genome-wide association studies where the frequency of common SNPs or variants across the genome are compared in schizophrenia cases compared to ancestry-matched controls. And there are a number of interesting results showing a modest association at particular loci. But again, these have not been replicated in most studies. And what I'll be talking about mainly today is the discovery of rare DNA variants. So what is DNA variation? I've split it into 3 different categories. Large cytogenetic aberrations that are 2 to 3 megabases or million base pairs in size and can be seen under a microscope. Copy number variation, or CNV, ranges in size from 1 kilobase to several megabases. And then single nucleotide variants and insertions and deletions, which are 1 to 25 base pairs. One of the most interesting cytogenetic aberrations in schizophrenia is a balanced translocation between chromosome 1 and chromosome 11. You can see here that the tip of chromosome 1 is replaced with the tip of chromosome 11 in a balanced translocation, so there's no loss of DNA during the translocation process. So this tip of 1 is transferred to the end of 11, and the tip of 11 is transferred to the end of 1. This was identified in a very large Scottish kindred in the early 80s, and that family's now being followed for the last 30 years with psychiatric evaluation. The breakpoint at chromosome 1 and chromosome 11 was completely revealed by sequencing, and the breakpoint at chromosome 11 is actually in the gene desert, but in chromosome 1 it disrupts a gene called DISC1, disrupted in schizophrenia 1. When the gene was identified, it was a novel gene with motifs suggesting that it might be involved in protein-protein binding. And since then, it looks like DISC1 is actually acting as a hub, linking different functional complexes within the brain. Interestingly, also at the chromosome 1 breakpoint is another gene called DISC2, and it's actually on the other strand of DISC1, and it's a non-coding RNA. And lots of people speculate that this is actually the target of the translocation, but it hasn't been fully revealed yet. This is a pedigree of the family with the translocation. You can see it's a 5-generation pedigree now. People that have inherited the translocation in a dominant manner have the red arrow. And as of 2001, there are 7 individuals diagnosed with schizophrenia, 10 with 10 with recurrent major depression, and 1 with bipolar affective disorder. So of the translocation carriers, the 29 carriers, 18 of those have a major mental illness. And of the non-translation carriers, none of them have major mental illness. So you can see that it's explaining some of the mental health in the family, but not all of it. And that's kind of a trend that you'll see throughout the talk, where there's an association, but there can still be people that are perfectly fine with the genetic mutation. And there's a variability associated with the mutations too. So getting on to copy number variation. They can be described as losses or gains or deletions and duplications greater than about 1,000 base pairs. About 12 to 15% of the genome varies in copy number. They can be inherited or de novo and readily identified by array, CGH, or commercial SNP platforms. And over the last few years, there's been a big push to get common and rare CNVs onto databases, such as the database of genomic variants. And it really, CNV represents a genome-wide mutation discovery approach. you can look at anywhere in the genome with your array, CGH, or SNP platform and ask whether there's a deletion or a duplication. This is a summary of the studies involved in schizophrenia CNVs from 2000, 2008 and 2009. The first one was by Kirov. It's a study from Wales. They looked at 93 cases with BAC array CGH platform. We then looked at 150 cases, mainly from Western State Hospital in Lakewood, Washington, and ancestry and age match controls with no history of mental health. Then there was a family study by Zhu et al. That looked specifically at de novo CNVs, and then a real increase in case control studies, and 2 very large case control studies. The International Schizophrenia Consortium, that looked at almost 3,400 cases and 3,200 controls. And then this very large study by Stephenson et al. At 4,700 cases and 42,000 controls. So the main findings from the schizophrenia CNV surveys are that, firstly, there's a genome-wide burden of rare mutations. So if you look at the CNVs and you filter out all the common CNVs that are greater than 1% frequency, and you just specifically look at rare CNVs, and in this case, those that are greater than 100 kilobases, there seems to be a lot more in individuals with schizophrenia compared to controls. And that's been seen in a few other studies too. And what we saw was that those CNVs were more likely to impact genes involved in brain function. The second main finding was that de novo CNVs, the frequency of de novo CNVs is increased approximately 8-fold in patients from sporadic families compared with healthy controls. And finally, using those very large case control studies, it was readily obvious that you could observe recurrent mutations, and 7 copy number variants associated with a high risk of schizophrenia have now been identified, and most of them are replicated. This is a picture showing where the recurrent CNVs are located on chromosomes. There are 5 deletions, one at 1q21, one at chromosome 2p, which is actually just spans a single gene, neurexin 1, 2 on chromosome 15, q11.2 and q13.3, and then 22q11.2, which is the VCFS locus. There's also 2 duplications associated with schizophrenia, both on chromosome 16, p11.2 and p13.1. So for 6 of those recurrent mutations, there is a local genomic architecture that's driving the deletion or the duplication. And I'll take you through that. You can see in this little diagram there's unique sequence represented by A, B, C, and D. And A and B are in between A and B is a segmental duplication indicated in orange, and the same between C and D. Now, these regions are highly homologous, greater than 95% identical, and they act as substrates for non-homologous recombination or crossing over, and they represent genomic hotspots. It was first worked out by Bailey et al., looking genome-wide at where the hotspots are located. This was work done by Evan Eichler here at UW. So how does the genomic architecture lead to these recurrent CNVs? This is an example of a DNA fragment that's come close to another DNA fragment of high homology during meiosis. And during recombination, you can see that A recombines with D to loop out B and C. And the recombination occurs in between these orange segments of segmental duplication. So you end up with a deletion of the BC fragment. You can also have a reciprocal duplication where the recombination starts here at C and goes to B, C, and you end up with A, BC, BC, and D. So 2 copies of the BC fragment, resulting in a duplication. And when we look at an array CGH or a SNP platform, this is what that data will look like. There'll be a decrease in intensity of probes or BACs, indicating a deletion. and an increase in protobacs or intensity of SNPs, indicating a duplication. So the first recurrent CNV that I'm going to talk about is 22q11.2. This is the chromosome blown up, and it's at megabase 17 through approximately 20. This is the cytogenetic band, and these orange fragments represent those segmental duplications, or low copy number repeats, that mediate the deletion or duplication. The next track is the list of well-characterized genes within the region. And there are two relatively common mutations at this locus. One is the full 3 megabase, or 3 million base pair deletion that spans the entire region. And another one is a 1.5 megabase within that 3 megabase region. Both of these deletions give rise to velio-cardiofacial syndrome, which is also known as DiGeorge or 22q11.2 deletion syndrome. It's characterized by craniofacial, immunological, and cardiac defects. It occurs at approximately 1 in 4,000 births. And interestingly, about 25% of adult deletion carriers develop psychotic illness, which is generally schizophrenia, but can be autism in some cases. So using that large case control study from the International Schizophrenia Consortium, they were able to show that there's a huge enrichment of this deletion in cases, and in fact none in controls. They found the frequency at about 0.3% with a huge odds ratio. So the deletion takes out about 25 to 30 genes. Some of the candidate genes that might be associated with schizophrenia include COMT, which is a methyltransferase involved in regulating neurotransmitters. PROD-H was involved in proline metabolism, and TBX1, which is a transcription factor. The second recurrent mutation associated with schizophrenia is at 1q21.1. It's a 1.35 megabase deletion, and this is the data from both of those large case control studies. It was detected in 17 of almost 8,000 cases for a frequency of 0.2%, and about 1/10 less in controls, and frequency of 0.02%. So highly significant and a very high odds ratio for this mutation. And all of these genes are expressed in the brain, but there are no standout candidates for a schizophrenia or a mental illness gene. And this deletion is also associated with variable phenotypes. And a follow-up study, Mefford et al. From UW, looked at 5,200 patients referred for genetic testing with mental retardation. 25 of those carried the exact same deletion with the same breakpoints as the schizophrenia cohort. 21 of those 25 had no other chromosomal abnormalities. And most of the 21 carriers had mild to moderate developmental delay. A number had dysmorphic features, and approximately 2/3 had microcephaly. There was also heart abnormalities, hypertonia, seizures, and cataracts. And interestingly, 4 of the patients had a normal cognitive development. And about the same time, another study saw pretty much the same phenotype. They had a much larger population base, that's 16,000 patients referred for genetic testing with mental retardation. 27 of those carried that exact same 1.35 megabase deletion, and most of those had developmental delay and or learning disabilities. In this study, they also saw a high frequency of the reciprocal duplication. So instead of just one copy, they had 3 copies of 1q21.1. And they were able to show that the deletion was associated with macrocephaly, whereas the duplication was associated with, the deletion was associated with microcephaly, and the reciprocal duplication was associated with macrocephaly. The third region is on 15q11.2. This is a very small deletion, just less than 500 kb, and is a lot more frequent in cases and controls. It's pretty close to 1% in cases and 0.2% in controls. The odds ratio is quite small. It's only about 3. There is a really nice candidate gene within that deletion. It's called CYF1P1, which interacts with the Fragile X mental retardation protein and seems to be involved in developmental maintenance of neuronal structures. The fourth locus is a 15q13.3, and this also has a very high odds ratio. It's at about 2% in cases and a 0.02% in controls. And again, there is a nice candidate for schizophrenia. It's a CHRNA7, which is a nicotinic acetylcholine receptor and mediates fast signal transmission at synapses. This mutation is also associated with a variable phenotype. So again, work from Evan Eichler's lab showed that in 9 out of 1,000 cases with mental retardation and seizures carried the exact same deletion. This was followed up with a study of epilepsy, which showed the mutation was associated with epilepsy, idiopathic generalized epilepsy. And finally, it's also been shown to be associated with mental retardation and autism. 6 of the 14 cases in the final study had autism spectrum disorders. Going on to the chromosome 16 loci, this is the first of the duplications associated with schizophrenia. It's at about a frequency of 0.3 in cases and 0.03 in controls. And interestingly, the reciprocal deletion at the exact same breakpoints is found in 1% of individuals with autism and about 1% of cases with unexplained intellectual disability. So you're probably seeing a trend here where mutations look like they're associated with schizophrenia, but then they seem to also be associated with other phenotypes too. And then the final recurrent CNV is on chromosome 16p13.1. It's also a duplication and is only about 3-fold increased risk for cases. And one of the candidate genes in this region is ND1 that's involved in brain development, neuronal proliferation, migration, and synapse formation. And it's particularly interesting because it actually interacts with DISC1 back from the translocation family. So there are 6 CNVs mediated by segmental duplications that are recurrent and seem to be associated with schizophrenia and other neurological phenotypes. Neurexin 1 is slightly different. It's a single gene mutation, and there is no common breakpoint. And Neurexin 1 itself codes for a presynaptic protein that binds to postsynaptic neuroregulins. And vertebrate neurexins are the only adhesion molecules which have a specific synaptic function. We and others saw rare neurexin deletions in childhood onset schizophrenia and autism. And this is the data from the neurexin paper. This is a screenshot from the UCSC genome browser. This is the original mutation that we identified in childhood onset schizophrenia. And then the subsequent mutations are up here. The red ones are cases, and the green ones are controls. And you can see there is no distinctive breakpoint. They're scattered throughout that genomic region. And what the authors found were that CNVs that disrupt specifically exons seem to be enriched in schizophrenia, but the odds ratio was quite low and the p-value was also quite low. So just to summarize the recurrent CNVs, there are 6 mediated by segmental duplications. The odds ratio vary from 2 to 3 up as high as 20 for VCFS. There are definitely other associated disorders with each of these genomic rearrangements. And the 7th one is a single gene enrichment of neurexin. So those 7 recurrent mutations account for approximately 2 to 4% of schizophrenia, definitely have a variable phenotype and an increased risk for other neurodevelopmental disorders. And it looks like there must be shared pathways for schizophrenia, autism, and mental retardation development if those deletions and duplications are common to all 3 phenotypes. And then I think an interesting question that people will be trying to work out over the next few years is how is that variation modified? Is it unmasked recessive mutations on the remaining allele that's not deleted? Or is it epigenetic or transcriptional regulation of genes within the deletion and duplication? Or is it just genetic background? So how about the non-recurrent CNVs? CNVs that are unique to an individual with schizophrenia. They're not present in databases of healthy individuals and at a very low frequency. So we have many of these from our patients that we've studied over the years, and we focus on those CNVs that either disrupt or alter a function of neurodevelopmental genes. And I'm going to take you through one of the examples that involves the glutamate pathway. So glutamate is the predominant excitatory neurotransmitter in the mammalian central nervous system. It activates two classes of receptors, and the uptake is mediated by a family of SLC transporters, 1 through 5. I'll draw your attention to number 3 here, which is also known as GLAST, for glial specific transport. So this is one of those rare CNVs that deletes approximately 500 kb on chromosome 5. It was found in a patient that had childhood onset schizophrenia, diagnosed at age 13. Her parents were not available for testing, so we don't know if the mutation arose de novo or was inherited. It's mediated by line elements, which are basically small repeats of about 3 to 4 kb. And when we found the breakpoint, we noticed that it disrupted a gene called SCIP2 and intron 2, and in intron 3 of GLAST, the glutamate transporter. SCIP2 is a ubiquitin ligase complex protein. It has a wide range of tissue expression, and GLAST is the high affinity glutamate transporter. It's just restricted just to brain. So we were fortunate enough to have RNA from whole blood from the schizophrenia patient, and we also had control blood RNA. And the first thing we did was an RT-PCR, reverse transcriptase PCR of SCIP2, amplifying a fragment from exon 1 to exon 4. And you can see it's expressed in the individual with schizophrenia, the RNA, the blood controls, and a commercial source of brain RNA, human brain RNA. So we did confirm it was ubiquitously expressed. For GLAST, we did not see any message by RT-PCR amplifying from exons 2 to exon 5 in the schizophrenia patient or the blood controls, but we did see it in that commercial source of brain RNA, confirming that it is restricted to brain. So because of the genomic structure, we thought it was possible that there may be a fusion gene going from this non-deleted region to this non-deleted region. So I put an RT-PCR primer outside the deletion and amplified from exon 2 to exon 4 and got a product in the patient with a deletion that wasn't expressed in the controls or the brain RNA. I went ahead and sequenced that junction breakpoint and showed that it is in fact a fusion gene, with the first 2 exons of SCIP2 fused to exons 4 through 10 of the transporter. It actually remains in frame and just makes one amino acid at that splice junction. So this is an example where the function of 2 genes has been drastically altered to create a new protein. We don't know what it's doing in the patient, but presumably if this part is functioning, it's now expressed at all tissue types, not just the brain. So maybe a dominant negative type mutation. So one of the goals of our lab is to improve the detection resolution of CNV detection. At the moment, with array CGH and SNP platforms, it's limited to about 25 kb on a good day, but realistically it's about 50 kb. It's also limited to just finding loss and gain, so deletions and duplications, and it can't pinpoint complex rearrangements. So we've taken advantage of next generation sequencing technology, or massively parallel sequencing technology, that allows you to capture between 50 and 100 base pair reads, millions and hundreds of millions of times. And we're using an approach called mate paired sequencing, where we have a fragment of DNA that's sheared down to a specific size. We do multiple rounds of size selection to make sure it's a very tight distribution. In this example, it's a 3 kb. And then we sequence those three, at least 50 base pairs of those 3 kb reads from here and from here. And using that information, we can map it back onto the genome browser and ask where are there any deviations from 3 kb. In this case, it would represent a deletion. I'm sorry, in this case it would represent a duplication where the ends are closer than you would expect. And in this example, the ends are further away, indicating a deletion. I'll take you through some examples that we have. So we decided to do a paired end library from a known control individual, NA15510. It's been well studied in array CGH and genomic studies in general. And it's actually our reference genome for array CGH projects. So it seemed like a good genome to sequence. So this represents a series of reads from a region on chromosome 17. And each little tag box here is a 50 base pair read aligning to the genome. And usually they should be approximately 3 kb apart, but in this case, they're about 13 to 14 kb apart, suggesting a deletion has occurred in here. And each one of these reads can be used to finally map where that deletion is. So we sequenced across this breakpoint, and it showed us a deletion of 10,233 base pairs. So better than array CGH, but not much. We then pushed it a little further and looked at another region of chromosome 17 and looked at overlapping reads that indicated there might be a deletion, a very small deletion. So again, each of these represents a 50 base pair read. And looking at the smallest region of overlap, we thought there may be a deletion of about 300 base pairs here. We sequenced through it and found a 323 base pair deletion. So this represents a great improvement on array CGH technology. How about improving sensitivity? So we know that we can detect small deletions, but can we detect more complex rearrangements? So in this case, instead of using just the mapping information, we'll also use the orientation of the mapping too. So this is an example of a tandem duplication where AB is inserted beside AB. And if we look in our paired end data, there should be a fragment that will correspond to this breakpoint, and you can sequence across to identify that. The same with an insertion where GH is inserted beside AB. There will be a number of reads that span that region too. And finally, an inversion. AB, CD goes to AB, DC, and this will be mappable too with a mate pair library. So how about those single nucleotide variants, insertions and deletions? We now feel that this is where most of the variation is going to be in psychiatric illness. And we're lucky, being at UW, that Jay Shanduri has really pushed this technology forward immensely. And we're actually located quite close to his lab, so we're always tapping his brain and his lab folks for ideas. So it's now possible to resequence a targeted region, a large genomic region, pretty easily and pretty cheaply. And using Jay's technology, it's also feasible to enrich for all coding region mutations of the genome, which has become known as the exome, and sequence many individuals. And Jay and others have successfully used this to identify causative mutations in rare pediatric disorders. So the approach we're taking in our lab is to go back to those 6 recurrent CNVs. We know that they are associated with schizophrenia, so it's possible that there'll be other mutations within those genomic regions that may also be involved with schizophrenia or psychiatric illness. So we've targeted the unique sequence between each of those recurrent CNVs, and this is the rough gene count and the rough size in megabases. So not a huge project really, less than 100 genes and less than about 10 megabases. So what we've done is design an oligonucleotide array that will capture just those regions within the segmental duplications. And we can then hybridize that array to a prepared library from a schizophrenia individual just to enrich for those regions. We'll then put it on our next generation sequencer and look for mutations predicted to alter function. They could be within the gene or controlling regulatory elements of the gene, or upstream or downstream of the gene. And the final approach we're taking is exome sequencing. We have two strategies. The first is to look at early onset child cases where healthy parents are available to sequence to. And we'll sequence all three individuals in a trio, mom, dad, and the affected child, and then focus just on the damaging variants that arise de novo. And then the second approach is to look at severely affected families. In this case, we would sequence all the people in the family, but just look at the variants that co-segregate from dad to his two affected sons, and focus on the damaging variants that are co-inherited with the phenotype. So in summary, I think we're now at a stage where we think mutations predisposing to schizophrenia are highly penetrant. They can be individually rare, of recent origin, and specific to single cases or even families. Thank you.
Questions? Dr. Tate?
Very interesting. Just going back to the risk factors, I remember a study a number of years ago which showed that there was a variation in the risk depending upon the time of year at which the baby was born.
All right.
Has that been confirmed, or is that?
I haven't kept up with that literature actually, but I do remember seeing it, the winter effect.
In terms of the sort of population attributable risk, it was a very sizable fraction of the total risk.
I'm not sure if that held up.
Have the studies that have been showed relationships to either the father's age or just those famine ones, were there any sub-studies that actually looked at maybe adoption or leaving to see if it was actually, because assumedly those children would have also grown up with those parents that had done all those things, so maybe having an older father and growing up with an older father is a problem.
I'm not sure. It's possible it was done, but I'm not aware of it.
I guess a related idea, are there studies to look at the prevalence of these kinds of mutations and as men age, and are the changes reflected accumulating in their sperm as they get older?
There are groups that do single sperm sequencing and looking at meiotic recombination events and DNA damage related events. I think there was a study of men that had been in Chernobyl and their mutation rates in their sperm.
And are they the similar pattern of mutations?
I think they were just looking at mini satellites, so highly repetitive regions that have quite a high mutation rate anyway.
So the prediction would be that those cases whose fathers are older would be de novo mutations.
Right, yeah.
I mean, has that been tested or confirmed?
Not that I'm aware of, no. But yeah, they would bring in a new supply of mutations into the population, the older fathers.
And what's the breakdown again, or is it known just what percentage of cases are thought to be sort of new mutations and what are inherited?
From the Zoo study, it looked about 10% of cases.
Are new
De novo.
mutations.
Is there any correlation between the psychopharmacology and the mutations that are shown up?
I'm not aware of that.
So when we find these, as more information accumulates about how strong these are associated with schizophrenia, when we find these in neonates or 1-year-olds, which we are, and some of these genes, I've seen these CNVs just in the last week.
Really?
What would you propose we do at this point as far as reporting?
Gosh. I don't think you could report a result in a neonate.
It's a tricky issue.
Yeah, definitely. I mean, they're not fully penetrant. Their phenotype is so varied.
And sort of related to that, when there's a very well-characterized, highly penetrant CNV, say for schizophrenia or whatever disease, that's say a deletion, and you see a duplication or some other mechanism, what would you propose we do with that?
In general, the duplications tend to be milder and have more varied phenotypes, generally.
What if there's nothing known about that? This is what's coming up.
Right. I don't think you could report that either.
Yeah, it's tricky.
I guess you need the statistical evidence for those rare ones.
And is this family of mutations, do you think, specifically associated with neuropsychiatric illness, or do you think this is just a representative of?
No, I think it's representative. Yeah, it's seen in other phenotypes, lupus, resistance to HIV. It's been seen in lots of other phenotypes.
Any other questions? Let's thank Dr. Walsh again.
Thank you.